welcome back. This is part 2 of genetics and inheritance. Thank you so much. I really hope you enjoyed the first one and you kind of understand genetics more. So yeah, today we'll be continuing with more genetics because this is a relatively big topic and it also counts for a lot of marks in the exam. So yeah, don't forget to like this video. Don't forget to subscribe, to tell all your friends, to tell everybody to tune in. So yeah, let's get to it. Okay, the last video we did codominance and we're just going to continue with codominance, but now in blood groups. As you know, there are 4 blood groups in humans, and that is blood group A, blood group B, blood group AB, and blood group O. We will say this is the phenotype because that is what is expressed. But then it is controlled by one gene, which is the ABO gene, which has 3 alleles. And these are your 3 different alleles: A, B, and I, which is an allele for blood group O. So both of these, A and B, are co-dominant over each other. No one is more dominant over the other, but both of them are dominant over I because it is a recessive allele. So the genotype for someone with blood group A can either be or this one, remember we said it's recessive, so because it's recessive, it is not expressed in the phenotype. So this could be a genotype with someone from a group that has blood group A. And same thing with blood group B. This is when the person is homozygous for blood group B. And this is when the person is, B, that's a B. This is when the person is heterozygous for group B. If a person has blood group AB, they can only be heterozygous because remember we said they are co-dominant. The allele for A and the allele for B is equally dominant. So that is the genotype for someone group AB. And then O, if you have blood group O, it can only be homozygous because this allele is recessive. So that is group O. So those are the possible genetics for the genotypes actually for individuals with group A, group B, group AB, and group O. So now we're going to do a genetic cross because they do like to ask this, because this is also used in paternity. They can use blood groups in paternity tests. So yeah, let's just get to the genetic cross. Okay, question 17 seems like fun. It says, in a court case between a biological mother and a potential father, the judge decided that the paternity of the child should be determined. Blood tests of the biological mother, potential father, and the child resulted in A, B, and O, respectively. Use this information and predict the possible verdict of the judge. Motivate your answer. Okay, let's get to it. Okay, so this is not a typical genetic cross, but we're going to be using a Punnett square to answer this question. So they told us, let me just have a look here. They told us that the mother, okay, phenotype. Oh, phenotype. The mother You don't, this is just me making it easier. Mother has blood group A, father AB, and child has O. Child has O. So you have to now determine whether the father is the father. So, okay, we're going to write out the possible genotypes to help us. Okay, mother could, for A, okay, we already wrote that down. For A, it could either be the homozygous or the heterozygous. Okay, we're gonna use a Punnett. Okay, I feel like using a Punnett square would be a long way to do this, but I can already see the answer that the father is not the father. Reason being, to have blood group O, you would need 2 recessive O alleles. And since the mother hypothetically has 1 O allele, where did the child get the other one? Because if you're blood group AB, this is the genotype. There's allele A and allele B. There is no recessive allele O. So he's not the father. He is definitely not the father. Going to be doing 6 linked characteristics next. 6 linked genes. Yeah, I'm going to be skipping 6 determinations because it is relatively simple. There will always be a 50% chance that a couple will get a male, a female, irrespective of whether they already have 3 female children and all of that. And also another thing that you need to know is gender or sex is determined by the male, because a female always carries the X chromosome. Okay, so you also need to know that the X chromosome is larger, and because it is larger, it can carry more alleles and as opposed to the Y chromosome. So that is why it is more common for men to have sex-linked I won't call them diseases, but I would just say, sex-linked diseases such as your hemophilia, your color blindness, because they only have 1 X chromosome to work with. And so let me just show you. We're going to do, using an example with hemophilia. Okay, so hemophilia is a recessive gene. This is hemophilia. It's recessive. Hope I'm spelling that right. No, yeah, you get the picture. Hemophilia, and this is a normal blood clotting G. Okay, so the reason why I say it is easier for a male to have such, because a female would need 2 recessive alleles for hemophilia, whereas if a man just has 1, then he has hemophilia. It's done. So that gives us these following combinations. This is we'll just say an affected male. Affected. We'll just say normal. I don't really like using the term normal, because what is normal? This woman, she is a carrier. She doesn't have hemophilia, but she is a carrier because she has the recessive gene present, but it is not expressed in her phenotype because it is recessive. and then this is an affected female. So you see, it's far easier for a male to, the chances of males having hemophilia are higher than that of females. So we're just going to do a genetic cross to put all of this in action. Okay, question 28 is what we're going to be doing. Red color blindness is also a sex-linked disorder like hemophilia. A man with normal color sights in marries a woman who carries the gene for red-green color blindness. Answer the questions that follow. What is the probability that their sons or daughters might be red-green colorblind? Okay, let us do a genetic cross to be able to determine that. Okay, we're just gonna get our ducks in a row. So we have a man. Okay. P1. You write phenotype. I do not have the space to write phenotype, so just know that that stands for phenotype. You write phenotype. Okay, normal color sight. Normal, I'll just say normal sight because of space. Writing the permanent marker sucks. Crossed with a carrier. And then the genotype okay, meiosis, and then we have our gametes. Okay, fertilization. And then we do a Punnett square. Okay, G for gametes, and okay, we'll just, those go there. And that is the female's gametes. Okay, and then they cross. Will I even be able to get all of this in? Oh my word. A permanent marker is not ideal. Ooh, okay. Then we write F1 genotype. We'll just stop there, genotype, and then it is other one is you usually try and just fit all of this in a line, but you kind of see my situation. And then the phenotype. Okay, I just want to read the question. Probability that their sons/daughters might be, okay, red-green colorblind. So we have a normal female, a normal male, a carrier female, and boom, colorblind. So to answer the question, the probability of that is 25% probability of red-green color blindness. Well, let me just write that out. Color blindness. Okay, so we've answered that question. And then 28.2 says, what is the probability that their daughters will have normal color sight? The probability that their daughters will have normal color sight. Okay, I say it's 50%, although she is a carrier. In her phenotype, oh wait, so this is actually, yeah, in her phenotype, she sees normally, even though she's a carrier, but she has normal eyesight. So I say 50%. No, it's 25%. I don't know, it's 25%. And then what is the probability that their daughters will be carriers? Then that is where you say 25% that they will be carriers. And then the last question is, red-green color blindness more common in men than women? Yes, it is, because men only have one X chromosome, which is a carrier. So if they get the gene, they cannot be a carrier. They are affected. Yeah. So that is it. Okay, next up that we're going to be doing is pedigree diagrams. They are basically diagrams that show the inheritance of characteristics or a characteristics in families over several generations. So 1 and 2 would be referred to as P1. This is not straightforward. It's not like, it's not as simple, but no, actually it is simple if you just apply the techniques and if you use the key. If you use the key, it's going to be very easy to figure out what they're doing. Actual facts in this diagram, because the genotypes of all of the individuals are included, that makes it very easy. But in the typical exam questions that they will give you, you will just get the key with no genotypes, and then you have to sort of figure the genotypes out, either by working backwards, by working with what you have and all of that. And I'll just show you how you can do that. Okay, so I've drawn out the pedigree diagram, and I hope there's something that you notice that is missing. Firstly, this is just, I don't have a figure title, so I should actually have figure 1 pedigree, or yeah. So that's just aside. Whenever you draw a figure or something, just always give it a title. Pedigree diagram of, okay, and you underline that. Okay, anyways, moving on. The question says, what are Katie and Mark's genotypes? Explain. Okay, so what I would advise you when you try and figure out the genotypes from a pedigree diagram is to always fill out the recessive genotypes first. By recessive, I mean homozygous, in a sense where in this diagram, black is the recessive gene. So yeah, we're gonna start off with that, and I'll just show you what I mean. Okay, I'll just write there that brown and that capital tells us that is dominant, black recessive. So we start off with everyone with black hair, because in order to have black hair you need to be homozygous for it. So that's easy. So we look at our key. Everyone with black hair is the one with a clear, either a clear square or circle. So Jessica, that is Jessica's genotype. That is Justin's genotype. That is Mark's genotype. It's always wise to do that, even though the question just asked for Mark. was it Mark and Katie's, yeah, Mark and Katie's genotype. Just do everything at first and then you'll see how simple it is. Okay, and then afterwards you figure out that, okay, Sandra has brown hair, but we don't know whether she's homozygous or heterozygous, so we'll leave her for now. But Mark is homozygous and their offspring have brown hair, and the only way that was their genotypes is going to be capital letter B and small letter b. It cannot be capital letter B and capital letter B. They cannot be homozygous for the brown hair because they do not have another dominant allele from their father. Their father only has lowercase. So I hope you get what I mean by that. So we can fill that in. And then Martin is a male with brown hair. Okay, Martin gets tricky because we don't know if he's homozygous or heterozygous, because he could be either, you know. And there's also Julia, which we also don't know. No, we actually do know, because sorry about that, Justin is homozygous, so it's the same thing as well. They're going to be heterozygous because they do not have another capital B. They only have one, because Katie married Justin. You see, this means that they not necessarily married, but this shows fertilization or whatever, and this is the offspring. So yeah, so we've got everyone except Sandra, and to be honest, we'll never really know whether Sandra is homozygous or heterozygous, so we'll just say that is her possible genotype. So yeah, we've already answered the question. Okay, I made that question up on my head, so I forgot. I think I said Mark and Katie. So if we had to answer question 1, what is Mark and Katie's genotype, we say that Katie's genotype is capital letter B, small letter b. She is heterozygous, and yeah, she's heterozygous, and Mark is homozygous, lowercase b, lowercase b. So yeah, and then another possible question that I'm making up out of my head is, what is Martin's phenotype? Martin's phenotype, Martin is a male with brown hair. Yeah, because it's in the key. So yeah, I think that is it for pedigree diagrams. I hope I didn't confuse you. I really do hope I didn't confuse you. If I did, please like drop me a question and like just, you know, yeah, but I really do hope you understand how you do that. Okay, yeah, that is it for today. Thank you so much for tuning in. I really hope you did enjoy this video. There's only one part left of genetics, well, 2 parts: dihybrid crosses and mutations, that I will cover in the next video. So that is part 3. Yeah, genetics is really that big. So yeah, if you have any questions, please feel free to ask. Please, you know, just comment, be like, I didn't understand when you did this, and just let me know, because you really can do this. Thank you so much for tuning in. Again, don't forget to like, to subscribe, and just share with everybody who might need this. Thank you so much, guys. Bye.